Welcome to the Da Vinci Academy Histology video course. The entire video course is available on YouTube and covers all of the fundamental principles of histology and relevant cell biology. You can find all of the videos from the course by clicking the Histology playlist link in the description below, and then you can access the corresponding practice questions and histology lab videos by going to our website, which is also linked in the description below. So this is the first of 3 lectures covering connective tissue proper, which is actually one of the 4 main tissue types that we're going to go over in this first unit. And in this first lecture here for connective tissue, we're going to talk about what is connective tissue and then the fibers that comprise connective tissue. And the 4 basic tissue types again, so Dr. Lee talked to you about epithelial tissue. This lecture we're going to talk about connective tissue, which is a broad term that includes a number of different tissues. So obviously connective tissue proper. Connective tissue also includes cartilage. It includes bone, adipose tissue, and several other types of specialized connective tissues as well. So in this lecture, we're mainly going to focus on connective tissue proper because we have other separate lectures exclusively dedicated to these other types of specialized connective tissue. So what is connective tissue? It consists of extracellular matrix and cells. And what's important to remember about connective tissue is that there's a much higher proportion of the extracellular matrix than there are cells. Certainly there's cells floating around, but the major component of connective tissue are these supportive fibers like collagen, elastin, fibers like that. Versus in the epithelium, remember that this was reversed. The major component of epithelium is the epithelial cells, and then you do have some extracellular matrix components to support those cells. The main function of connective tissue is to support and protect the functional tissue of organs. This is a great segue into explaining the difference between parenchyma and stroma. So if we look at the submandibular gland here, this is a zoomed out view here. And if you look at it, the parenchyma is very easily defined. And so this is the glandular tissue here. And you can see it here again. And then if you zoomed in here again on this portion here, you can see it's very easily defined. If you look on the outskirts here, here's this eosinophilic fibers like this kind of surrounding it, separating these different functional units. This is what's called the stroma. So again, this is the parenchyma, which is a general term that just refers to the functional tissue of any organ. And then you have it here, so this is connective tissue here. And you can see out here, here's the, it's again, stains very eosinophilic, a lot of proteins, extracellular matrix. You can see some blood vessels in here. You see some adipose tissue in here, found right here. And this is what's called the stroma, which is the supportive tissue of the organ. So it supports the parenchyma. Most connective tissue elements originate from the mesenchyme, which is essentially embryonic connective tissue. And then as far as the germ layers go, remember you have the mesoderm. And the mesoderm gives rise to some connective tissue, but it also can give rise to epithelial and muscle tissues as well. So the connective tissue components, again, we said extracellular matrix and cells. Extracellular matrix is made of extracellular fibers, ground substance, and fluids. Specifically, it has collagen, reticular fibers, elastic fibers, glycoproteins, proteoglycans, and glycosaminoglycans, or GAGs. And we'll talk about each of these individually over the next few slides. Cells, main cell you're going to find in connective tissue proper, especially is fibroblasts. You can also see adipocytes in adipose tissue. You'll see white blood cells floating around contributing to immune function. And you can also see mast cells as well. And so if you look at this is a great example of what's called loose connective tissue, and we'll explain more in detail what that means over the course of the lecture. But just to give you a sense here, you can see there are some cells in here. Here's a mast cell here. Here's some other cells floating around in here. But you'll notice that it's not mainly composed of cells. There's a lot of other fibers here. So this would be an elastic fiber here. You can see these red squiggly lines like this. These are what I would call collagen fibers, because remember again, proteins often will stain eosinophilic. And so you see a lot of different fibers. And then you kind of see this what's called ground substance, which is this gelatinous-like substance. And it's almost like a soup, if you will. It's you have the ground substance, you have some fluid in there, some water component to it. You have a lot of these fibers floating around, and then kind of the component of the soup, if you will, you have these cells floating around within it. So the most prominent structural protein of the extracellular matrix is collagen. And it's also the most abundant protein in the human body, which isn't too surprising because you have supportive connective tissue all throughout your body and literally almost every organ. And the reason for that is that collagen provides significant tensile strength with limited flexibility. So it's a great supportive fiber. And there's 4 major types that you need to be familiar with. There's type 1, type 2, type 3, and type 4. And so these are just some images. We'll revisit these again throughout the lecture, but just to kind of point out some collagen fibers, kind of in black here, these would be supportive collagen fibers within the lymph node. These are also known as reticular fibers. Talk about what that means in a second here. Then if you look at this slide from skeletal muscle, so this is the skeletal muscle tissue down here in kind of this purplish reddish color here. And then here, these bright red fibers here, these are collagen fibers. And so you can see where they stain very brightly red usually. So the different types, this is just kind of a table to help you figure out the location and the function. It's really important to know both the location and the function for each of these. These are very commonly test questions. So you have collagen type 1. So type 1 is very strong. It's able to sustain a large amount of force. Therefore, it's found in bone. It's actually synthesized by osteoblasts within the bone, but it can also be found in the tendon, dentin, fascia, and then the cornea within the eye. And what's important to know with collagen is that you have the collagen protein, which then forms collagen fibrils by cross-linking the proteins. And then those merge together to form collagen fibers. And we'll go over this when we talk about collagen synthesis in a few slides here. The other thing is that what that's important to know that's very high yield for exams, especially a board exam, is that collagen type 1 is produced during the late phases of wound repair to form a fibrous scar tissue. So just a very brief overview of wound repair. So if you have a wound like this, kind of a break in the skin, the first thing you're going to do is, obviously you're going to be bleeding. So the body's going to achieve hemostasis by forming a clot in here. Then you're going to have, cellular debris because it's essentially damage. So you got to have these immune cells come in, essentially clean things up, the cleanup crew. Then next, what you're going to do is you're actually going to lay this kind of structural framework or this scaffolding. And these are actually collagen type 3 fibers or reticular fibers. And they kind of serve as the framework for then later stages where you actually form the fibrous scar tissue by laying in your collagen type 1 fibers. And that makes sense. Collagen type 1 is much stronger. It's going to give you a much better much stronger scar tissue here to help fully seal this wound. So you go from collagen type 3 to collagen type 1. Again, very commonly a test question on the boards. Type 2 collagen is found in cartilage and in the vitreous body in the eye. It's also found in intervertebral discs. Specifically, if you remember a disc structure, it's kind of like a jelly-filled donut. You have this kind of gel in the middle here, and then you have this outer surface here, this much tougher kind of structural surface here. And this inner portion is called the nucleus pulposus. This is composed of type 2 fibers within this gelatinous mix in here. Then you have the annulus fibrosus, which is this much more tensile, much stronger covering around the gel. And this is composed of both type 1 and type 2 fibers. Type 3, as we say here with the star, is also called reticular fibers. And these, again, they kind of cross-link like this. These fibers will come together and they'll cross-link like this to kind of form a scaffold or a mesh structure. And they form in a lot of different areas. The skin, mainly the dermis, blood vessels, lymph nodes, the uterus, and fetal tissue as well. They're also, as we said, produced during the early phase of wound repair to produce what's called granulation tissue. So that framework that's laid down first is called granulation tissue, composed of type 3, and then you form on top of that the fibrous scar tissue, which is composed of type 1. Then lastly here, type 4. This forms flexible fibrous sheets found in the basal lamina and the basement membrane. So again, if you have your epithelial layer like this, and so you have your lumen out here where absorption is occurring or secretion, and then you're going to have this basal lamina or basement membrane where all these cells are adherent to, and this is mainly composed of collagen type 4. These are just some histological examples. So this is from the bone, this is type 1, so you can see that in here, very compact structure. This is type 2, which is cartilage. Type 3, again, this is from a lymph node. These are those reticular fibers. You can kind of see them here in black. And then type 4 here, this can be difficult to see. It's really what you're just kind of looking for is this line here that's forming uniformly below this epithelial layer, which is where you would see collagen type 4. So collagen structure is a pro-alpha chain, which is composed of the following sequence: glycine plus 2 more amino acid residues, and those are either proline or lysine. So it could be something like this, where you have proline like this, and then it repeats again like this, and it could be the reverse like that, and then so on and so on. And so as you can see, a good portion, actually a third of collagen, is glycine. And then this sequence is modified in the cell to form a triple helix composed of 3 pro-alpha chains. So you have 3 right here, 3 alpha chains, which then form a triple helix. And you can see this whole structure here is a triple helix. So pro-collagen triple helix, which would be this structure here, is actually exported outside of the cell, where it undergoes further modification to enable cross-linking between these triple helices. So you can see here's one here, here's another one here, and you can see these cross-links being formed here. And so the final collagen fiber is actually a structure with multiple cross-link collagen triple helices, and you can see that here. So here's triple helices here, cross-linked here, cross-linked here. It's synthesized by fibroblasts, which are cells found in connective tissue. So really the outline is you have the pre-pro collagen, which is synthesized within the cell, within the rough ER actually specifically. Then you have the pro-collagen, which is formed after some modifications to the initial sequence. That's exported out. And then you have tropocollagen, which is here. And then these undergo that cross-linking to form collagen fibers. So first, there's an error in this figure. This is not the cytosol. This is the extracellular space. This is the cytosol here. And actually, a good portion of this actually takes place within the rough ER, or the rough endoplasmic reticulum. And what that enables is actually exocytosis of pro-collagen, which would be this last portion here, to the extracellular space here. And then that's where synthesis is completed. So first, pre-pro collagen is translated from collagen mRNA in the rough ER to produce the pro-alpha chain, which is that glycine plus XY. And remember, one of these is either a proline or a lysine. And so that's just here, just like how any other protein is formed. Is first it's translated from the mRNA. So you have this initial sequence here. And then the proline and lysines undergo hydroxylation in the rough ER. And this is actually a process that requires vitamin C. Then after hydroxylation, they undergo glycosylation, as you can see here and here. And what these processes, specifically hydroxylation, do is they enable the helical alpha chains to spontaneously form a triple helix in the rough ER. And you can see that here, and this is what's called procollagen. And this occurs via hydrogen and disulfide bonds to help form this triple helix structure here. Then this procollagen is exported via exocytosis to the extracellular space, where it undergoes the final steps in collagen synthesis. So outside the cell, again, the terminal disulfide-rich propeptides of the procollagen, so this is procollagen here, are cleaved by peptidases to form tropocollagen. So you have cleavage here, and you can see these little ends coming off here. Then you form one of these triple helices with the C and N termini cleaved off, and this is tropocollagen. Then you have an enzyme called lysyl oxidase, which catalyzes the oxidation of lysine side chains of tropocollagen to yield reactive aldehydes. Then these reactive aldehydes form covalent lysine hydroxylysine crosslinks, and that's what is shown here between these tropocollagen molecules. And this allows for spontaneous formation of the cross-linking between the tropocollagen. So here's a tropocollagen cross-linking here and here to this one, and then it's cross-linking to this one as well. And so what ends up happening is that you have multiple tropocollagen fibers that are cross-linked together to form the collagen fiber. Cross-linking between tropocollagen chains within collagen fibers actually continues to occur, so you continue to get more of these cross-links throughout life, and it leads to increased stiffness of structures that contain collagen with age. particularly skin, joints, and arteries. And this is what leads to some of the detriments of older age. So over the next few slides, we'll talk about elastin and elastic fibers. So elastin is a highly elastic protein that allows tissues to be stretched and resume their native shape. So you have some kind of deformity on this or some stretch and shape, and then it resumes back to the native shape. And this elastic nature often contributes to the function of tissue that actually contains elastic fibers. So lung tissue, for example, contains a lot of elastic fibers because the elastic fibers allow the lung tissue to expand on inspiration, and then the recoil of the elastic fibers allows to push air out during expiration. And then the aorta also contains numerous elastic fibers because that's also contributing to its function as well. So if we look at the aorta during systolic ejection, ventricular ejection, it expands in all directions, and its elastic fibers allow it to do that. So it's able to expand and accommodate a higher volume of blood, and then when ejection ends, you have this diastolic recoil, which then gives you kind of an extra pulse of blood, or the diastolic pulse of blood, and that contributes to your diastolic blood pressure. Elastic fibers are also found in large blood vessels other than the aorta, such as the veins, the vena cava. It's also found in elastic ligaments, vocal cords, ligamentum flavum, which is a ligament in the spine, and then the bladder as well. So a lot of structures here that have to undergo a lot of dynamic stretching and expansion that are key to their function. Come down in this section here. This is from loose connective tissue. You can see this thready-like structure here. This is an actual elastic fiber where the arrow is pointing. Come over to this section here. This is an elastic artery. And then if you look in this section here, this is the muscular layer of the artery. So if we draw a cross section here of the artery, you have the endothelial layer, which faces the lumen. Then you have the muscular layer like this. And then you have the adventitial layer. And we'll go over in more detail in this in the cardiovascular lectures. But this muscular layer here is what contains the elastic tissue. And you can actually stain for elastin using an elastin stain. It's just in the name. And you can see these long, dark fibers. I'm trying to underline them here so you can see them. They're very numerous, as you can see, throughout this muscular layer. And again, that contributes to its ability to expand and then resume its native shape. Synthesis of elastin, like collagen, is synthesized by fibroblasts. And so the synthesis is very similar to collagen, and a lot of the terms are very similar. So you have within the cell here, and then you have outside the cell here. And tropoelastin is synthesized within the cell, and then it undergoes exocytosis to go outside the cell. And then outside the cell, you have lysyl oxidase again, which catalyzes oxidation reaction of lysine residues to generate these reactive aldehydes and allicin, which enable tropoelastin proteins to cross-link and form elastin. And so if you have a couple different tropoelastin molecules like this, lysyl oxidase allows them to cross-link like that, and that's what forms elastin. So you have tropoelastins, which then cross-link to form elastin, similar to tropocollagen fibers, which cross-link to form collagen fibers. Elastin is broken down by elastase, so this is broken down by the enzyme known as elastase, and then alpha-1 antitrypsin inhibits, and this can be important in a disease we'll talk about in the clinical pearls section. Elastin contains a high amount of non-hydroxylated proline, glycine, and lysine residues. So the elastic fiber, so once you have elastin, then you need to create the elastic fiber. So these are produced by fibroblasts and arterial smooth muscle cells, so to help contribute to that flexibility of larger arteries. So you have fibrillin, which is a glycoprotein that's synthesized and secreted by fibroblasts in the extracellular matrix. And so we'll draw just kind of a cartoon here. So this will be your fibrillin here. So this will be a glycoprotein that was secreted by the fibroblast. And then the fibroblast would have also secreted the elastin, which then is cross-linked like this. And so fibrillin serves as a major component of the microfibril scaffold for deposition of elastin. So you have the fibrillin molecule here, and then these cross-linked elastins come down and they deposit onto the fibrillin. like this, and then they can come on this side too. And so these neighboring elastin molecules, they aggregate on here onto the microfibril scaffold, and then they further cross-link with each other like this. And what happens is this completes the composition of the elastic fiber. So the elastic fiber is both the elastin and the microfibril scaffold, which is the main component of that is this fibrillin here. So all of this is elastic fiber. So elastin has a further component to it. So you create the elastin, which is these cross-linked tropoelastins here. Then they aggregate on fibrillin and then further cross-link and aggregate together, and that's what forms your elastic fiber.
Thank you for watching this video from the Da Vinci Academy Histology video course, which is completely available on YouTube. To access the corresponding practice questions and histology lab videos, go to our website using the link in the description below.